Imagine a world free from genetic diseases, a world where inherited disorders are a thing of the past, and every child is born healthy and free from genetic ailments. Imagine crops engineered to resist climate change, crops that can thrive in extreme weather conditions, ensuring food security for future generations, even in the face of global warming. This is the promise of CRISPR, a revolutionary gene editing technology poised to transform life as we know it. With CRISPR, we can precisely alter DNA, opening up possibilities that were once the realm of science fiction. CRISPR gives us the power to rewrite the code of life, to correct genetic defects and enhance desirable traits. This means not only curing genetic diseases, but also potentially enhancing human capabilities and longevity. However, this power comes with profound ethical questions that we must confront. The ability to edit genes raises concerns about unintended consequences and the moral implications of altering human DNA. CRISPR's potential benefits are undeniable, offering hope for treating diseases and improving food security. It could revolutionize medicine, agriculture, and even our understanding of biology itself. But like any powerful tool, it can be misused. The possibility of creating designer babies or genetically modified organisms with unforeseen impacts is a real concern. We must proceed with caution, engaging in open and informed discussions about the ethical implications of this groundbreaking technology. Public engagement and transparent research are crucial to navigating these complex issues. The future of gene editing, and indeed the future of life itself, depends on our wisdom and foresight. How we choose to use CRISPR will shape the world for generations to come. CRISPR's origins lie not in some high-tech lab, but in the humble bacterium. It all began with the simple observation of how bacteria defend themselves against viral attacks. Scientists discovered that bacteria use CRISPR as a defense mechanism against viruses. This discovery was groundbreaking, revealing a natural process that had been occurring for billions of years. These repeating DNA sequences found in the bacterial genome act like a cellular immune system. They are essentially a genetic memory bank, storing information about past viral invaders. They store fragments of viral DNA, allowing the bacteria to recognize and destroy invading viruses upon subsequent infections. This mechanism is incredibly efficient and precise. This remarkable system, harnessed and refined by scientists like myself and Emmanuel Charpentier, has given rise to a revolutionary gene editing tool. Our work has transformed a natural bacterial process into a powerful technology. CRISPR, paired with the Cas9 enzyme, can be programmed to target and cut DNA at specific locations. This precision is what makes CRISPR so revolutionary. This breakthrough allows scientists to edit genes with unprecedented precision, opening up a world of possibilities in medicine, agriculture, and beyond. From curing genetic diseases to creating crops that can withstand harsh conditions, the potential applications are vast and transformative. CRISPR holds immense promise for treating a wide range of diseases. Scientists are exploring its use in correcting genetic defects that cause diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. By targeting and correcting these faulty genes, CRISPR offers the potential for cures, where previously there were only treatments. Beyond treating inherited diseases, CRISPR could also revolutionize the fight against cancer. Scientists are investigating ways to use CRISPR to enhance the body's own immune system, enabling it to recognize and destroy cancer cells more effectively. This approach holds the potential to develop more targeted and effective cancer therapies with fewer side effects. The ability to edit the human genome, once the realm of science fiction, is now within our grasp. This raises profound ethical questions about what it means to be human and who gets to decide how this technology is used. While the potential benefits of CRISPR are enormous, so too are the risks. One of the most contentious issues is the potential for germline editing, altering the genes passed down to future generations. While this could potentially eliminate inherited diseases, it also raises concerns about unintended consequences and the possibility of creating unintended changes in the human gene pool. The spectre of designer babies enhancing versus healing. The prospect of using CRISPR to enhance human capabilities, creating so-called designer babies, is particularly fraught with ethical concerns. While the idea of enhancing intelligence, athleticism, or physical appearance may seem appealing to some, it raises serious questions about equity, justice, and the very definition of human potential. The line between therapy and enhancement is often blurry. Should we use CRISPR to treat serious diseases, but not to enhance height or muscle mass? Who gets to decide what constitutes a disability or a desirable trait? These are complex questions with no easy answers, requiring careful consideration and societal debate. CRISPR's uncertain future, navigating the risks. CRISPR technology is still in its early stages of development. While it holds immense promise, it is crucial to acknowledge and address the potential risks associated with its use. One of the primary concerns is the possibility of off-target effects, where CRISPR inadvertently edits genes other than the intended target. These unintended edits could have unpredictable and potentially harmful consequences. Scientists are working to improve the precision and safety of CRISPR, but it is essential to proceed with caution and rigorous testing before widespread clinical applications are considered. CRISPR and society, a call for open conversation. The ethical dilemmas posed by CRISPR are too complex and far-reaching to be decided by scientists alone. This is a conversation that must involve everyone: policymakers, ethicists, religious leaders, and the public at large. We need to engage in open and informed dialogue about the potential benefits and risks of this powerful technology. Public education is crucial. People need to understand how CRISPR works, its potential applications, and the ethical questions it raises. Only through informed public engagement can we hope to develop ethical guidelines and regulations that reflect our values and aspirations. The global landscape of CRISPR governance. The development and use of CRISPR require careful governance and regulation to ensure responsible innovation. However, the current regulatory landscape is fragmented and inconsistent across different countries. This patchwork of regulations creates uncertainty and hinders the development of safe and ethical applications of CRISPR technology. There is a pressing need for international collaboration and harmonization of regulations to address the global implications of CRISPR. A universal framework for the ethical and responsible use of gene editing is essential to prevent misuse and ensure that the benefits of this technology are shared equitably. A shared future, balancing innovation and responsibility. As we venture into the uncharted territory of gene editing, we must proceed with both audacity and humility. We must embrace the transformative potential of CRISPR while recognizing the profound ethical responsibilities that come with it. The choices we make today will have far-reaching consequences for future generations. We must find a balance between fostering innovation and safeguarding our values. This requires ongoing dialogue, ethical reflection, and a commitment to transparency and public engagement. Only through a shared sense of responsibility can we ensure that CRISPR is used for the benefit of humanity and the planet. CRISPR's legacy, shaping the future of life. This groundbreaking technology has opened up new frontiers in science and medicine. CRISPR has the potential to reshape the world around us, from eradicating diseases to transforming agriculture. Imagine a world where genetic disorders are a thing of the past and crops are more resilient to climate change. It is a powerful tool that can be used for good or ill. The ethical implications are vast and complex, requiring careful consideration and global cooperation. The legacy we leave behind will depend on the choices we make today. Our decisions will shape the future for generations to come, influencing the very fabric of life itself. It is our responsibility to ensure that CRISPR is used wisely and ethically to alleviate suffering and to advance human health. We must strive to balance innovation with caution and to create a more just and equitable world. Collaboration and transparency are key to achieving these goals as we work together to harness the power of CRISPR for the greater good. The future of life itself may depend on our ability to navigate the ethical challenges posed by this revolutionary technology. The stakes are high, and the potential for both positive and negative outcomes is immense. Let us proceed with wisdom, courage, and a commitment to ethical principle. We must be vigilant and proactive in addressing the moral dilemmas that arise, and a deep sense of responsibility for the well-being of all. Together, we can shape a future where science and ethics go hand in hand, ensuring a better world for everyone.
Hit that like button, subscribe, and stay informed.